Good morning or good afternoon everyone, and in this video we will be covering our drug concentrations and our pharmacokinetic models. So this is the second topic in the course Biopharmaceutics and Pharmacokinetics, and we will be dealing with the sources of our drug concentrations in the body and also what pharmacokinetic models are, the different types, and what they are useful for. So let's start with our drug concentrations. So in measurement of drug concentrations, these are an important element in determining individual or population pharmacokinetics. In particular, these are important for getting measurements of levels in our patients or in our population to establish kinetic parameters. So drug concentrations are measured in biological samples such as milk, saliva, plasma, and urine. So these are the most common sources of our drug concentrations in the body. And in general, chromatographic and mass spectrometric methods are used in drug concentration measurement. So we use high-performance liquid chromatography, gas chrom, and mass spectro since they are the instrumentation methods that could give us a measurement of concentration. It means that other methods such as infrared spectroscopy or our AAS cannot be used in measuring concentrations of our drugs, especially in our plasma. So when sampling biologic specimens, only a select type of sample may be used in analyzing our drug concentrations. We have two primary divisions of our methods of sampling. We have our invasive methods and we have our non-invasive methods. When we say invasive methods, these are methods that could need puncturing of the skin or any surgical intervention. So samples of this is your blood collection, sampling of spinal fluid, tissue biopsy, or any material that requires parenteral or surgical intervention. For our non-invasive methods, these are those that need not penetrate the skin. So these are usually our waste products from the body, like sampling our urine, saliva, feces, expired air, or any material that does not need puncturing of the skin or any surgical intervention. So when sampling biologic specimens, it is important that our analytical method should be able to distinguish between protein-bound, unbound, parent drug, each metabolite, and pharmacologically active species. It's not good or it's not ideal if the only measurement we can get from our drug analysis is the amount of drug in the sample. We need to know how much is bound and unbound, because our bound drugs in the plasma proteins, these are inactive. They may yield a certain concentration when sampled, but these are actually inactive. They do not contribute to the pharmacologic activity of the drug inside the body, particularly because they are bound to plasma proteins. When these are unbound or are free, these are the active species. So we need to know how many percent is bound and unbound, since only the unbound percentage actually contributes to the pharmacologic action of the drug. Next, we also need to know the parent drug and the metabolite, so we could gauge how much of the drug is actually metabolized or how far do we need to go to have a complete removal of drug from the body. And finally, it should be also able to identify pharmacologically active species from metabolized products which are pharmacologically inactive. So first is our drug concentrations in the blood, plasma, or serum. In general, serum or plasma is used for most of drug measurements. Please do take note that plasma and serum do not contain the same proteins, and the most important sampling or the most important sample that we need to obtain is actually from the plasma. It's because of these two reasons. Plasma perfuses all tissues of the body. It means every part of the body has plasma. Plasma moves everywhere inside the body, and it is a good representative for the whole body. And assuming that the drug in the plasma is in dynamic equilibrium with the tissues, if we take a drug sample from the plasma, it represents the whole body. So if the plasma concentration is 12 milligrams per liter, then the body tissues also have 12 milligrams per liter. In short, since the plasma is in dynamic equilibrium, what the concentration of the plasma is, it is equal to the drug concentration in the tissues. So that's the purpose of dynamic equilibrium. Tissue concentration equals plasma concentration. And this is important since we don't usually get tissue concentration, we only get plasma concentration. And when we get plasma concentration, we also get tissue concentration since the plasma is in dynamic equilibrium. Next, we have drug concentration in the tissue. So these are usually performed through tissue biopsies. So there is a surgical procedure involved wherein we will get a certain part of the affected area and we're gonna sample how much drug is in it. So tissue biopsies are occasionally removed for diagnostic purposes, such as for verification of malignancies. Tissue concentrations in the drug concentrations in the tissues may not be reflective of other tissues. So take for example, breast tissue may not have the same amount of drug as in other parts of the body. And the measure of drug concentration in the tissue biopsy material may be used to ascertain if the drug reached the tissues and reached the proper concentration within the tissues. We don't usually get biopsies just to get drug concentrations. However, in certain research scenarios and when we are verifying if the drug has reached the tissues and it is reaching an appropriate level in the tissues, then tissue biopsies are the way to go. But other than that, in regular drug concentration testing, we don't usually use tissue biopsies. Next is tissue concentration in urine and feces. So urine drug concentration is an indication of bioavailability. So it's an indirect method to ascertain bioavailability. Why is that? It's because for a drug to appear in urine, it must have first appeared in the kidney. And before it could have reached the kidney, it should have reached the blood first. So if the drug was able to reach the blood, therefore the drug was absorbed. So presence of drug in the urine means that the drug was absorbed. So it's a proof of bioavailability of the drug. For our feces, measurement of drug in the feces may reflect that the drug has not been absorbed after an oral dose, meaning if you take a drug orally and the drug appears in the feces, it means there is residue of the drug in the feces. It went through the GI tract without being absorbed. Drug in, drug out in the feces, meaning there is poor absorption of the drug. Or another case may be is that the drug is being expelled through biliary secretion after systemic absorption. We do have some drugs that are not excreted through the kidneys via urine but excreted through the bile. So the bile is a surfactant that helps digest fats. Sometimes drugs are excreted through that way. So if you find drug concentration in urine and feces, it may mean either the drug is poorly absorbed through oral route or the drug is being secreted via biliary secretion. Next, drug concentrations in the saliva. Saliva only excretes free drug. So you can only find free drug in the saliva. Bound drugs do not appear in the saliva. Therefore, if you sample saliva, you would only get free drug concentrations. And it appears, and saliva drug levels tend to approximate free drug rather than total plasma concentration. The use of salivary drug concentrations as a therapeutic indicator should be used with caution and preferably only as a secondary indicator of bioavailability. So again, let's reiterate why plasma is the best drug sampling site. So the intensity of the pharmacologic or toxic effect of a drug is often related to the concentration of the drug at the receptor site. Since most of the tissue cells are richly perfused with plasma, measuring the plasma is a responsive method for monitoring the course of therapy. Monitoring the concentrations of drugs in the plasma ascertains that the calculated dose actually delivers the plasma level required for the therapeutic effect. So that is why plasma is used in most of our charting of drugs. So next on hand, we have our pharmacokinetic drug models. So drugs are in a dynamic state in the body as they move between tissues and fluids, bind with plasma or cellular components, or are metabolized. So drugs do not stay in one place, but rather they move throughout the body. And the processes of Ladmer often happen simultaneously. It's not you need to finish absorption first, then move to distribution. Drugs undergo Ladmer processes simultaneously. So as the drug is being absorbed, it's being taken away to other parts of the body. As they are being taken away to other parts of the body, they are also being metabolized and subsequently excreted. So pharmacokinetic models allow a more accurate interpretation of the relationship between the plasma drug levels and pharmacologic response. Monitoring of the plasma drug concentration allows for the adjustment of the drug dosage in order to individualize and optimize therapy for drug regimens. So what's a model? So a model is a hypothesis using mathematical terms to describe a quantitative relationship concisely. So you might have already observed what different models are. We have economic models, we have models to show predictions. And the type of model we use here in biopharmaceutics and pharmacokinetics is called a pharmacokinetic model, since it uses pharmacokinetic parameters. So a model is a predictive apparatus. So if you have the appropriate background data, you could construct a model to predict situations in which you have prior data of. So a pharmacokinetic parameter is a constant for a drug that is estimated from an experimental data. So pharmacokinetic functions relates an independent to a dependent variable, often through the use of parameters. So pharmacokinetic parameters, we will be discussing them in our pharmacokinetics at the second half of our course. So pharmacokinetic models are used to the following: predict plasma, tissue, and urine drug levels, calculate optimum dosage regimens, estimate the possible accumulation of drugs, correlate drug concentrations with pharmacologic or toxicologic activity, evaluate differences in the rate or extent of availability between formulations or when testing bioequivalence, describe how changes in physiology or disease affect the Ladmer processes, and also it can be used to explain drug interactions. So, pharmacokinetic models are made by simplifying assumptions. When we have a pharmacokinetic model, it is an oversimplification of a complex biological microorganism concerning the movement of drugs within the body. For example, most pharmacokinetic models assume that the plasma concentration reflects the drug concentrations globally inside the body. We assume that plasma has a one-to-one correspondence of drug concentrations in the plasma and the different compartments of the body. We have several types of pharmacokinetic models. First, we have our empirical model. So an empirical model is prepared by collecting pharmacokinetic data from a population set. This is purely based on statistics and math, and they can predict various outcomes based on the data you put into it. However, the disadvantage of an empirical model is since it's only based on math, you cannot explain drug interactions or accumulation using empirical models. It's good for predicting concentrations, but it's not good for explaining things. A physiologically based model, on the other hand, takes into consideration more data, more clinical data, and it can consider all aspects of the Ladmer process to get the model. The physiologically based model is usually used in research to explain different phenomena when dosing a certain drug. And next, we have what we use in clinical practice. It is a compartmental based model. So it's a very simple and useful tool in pharmacokinetics. It takes into consideration the concentration of the drug, the volume of the compartment, and the rate of elimination. It is a model describing drug absorption, distribution, and elimination in the body, which is represented as one or several compartments. The compartmental modeling of pharmacokinetics consists of describing the fate of drug in the body divided into compartments. So a compartmentally based model tend to look like this. It is represented by boxes in which the body is separated or divided into different compartments. Take for example, you have an oral dosing. You represent the GI tract as one compartment. And the central compartment is your blood, since all drugs would end up in the blood anyway. And since the blood flows everywhere, it could go to other organs like the brain or the heart, or even to excretory organs such as your kidneys. So in the compartmentally based model, we separate the different parts of the body as different compartments. And the relationship between each compartment can be represented by pharmacokinetic constants such as distribution constant or elimination constant or absorption constant. So from GI to blood, it is an absorption constant. Kidney to urine, it is an elimination constant. Okay? It is a very simplified method of showing drug flow in the body, but it's also very effective. And it's one of the very useful models used in pharmacokinetics. A mammillary model is the most common compartment model used in pharmacokinetics. The mammillary model is a strongly connected system because one can estimate the amount of drug in any compartment of the system after the drug is introduced to a given compartment. So what I drew earlier is an example of a mammillary model. There is a central compartment, which is your blood. Drug enters the blood, blood goes everywhere, and it explains how the drug flows from one compartment to another. So here are various compartment models. You also have a quaternary model. So a quaternary model is like a train. The quaternary model consists of compartments joined to one another like the compartments of the train. So it is different from our mammillary model in which there is a central compartment. In the quaternary model, we simply assume that the flow of drugs is two-dimensional. It goes to one compartment to the other to the next, and we do not assume that the drug goes to other parts of the body. Usually, GI, blood, elimination for quaternary model. And finally, we have our physiologic pharmacokinetic model. So these are also called your blood flow or perfusion models and pharmacokinetic models based on known anatomic and physiologic data. These models describe the data kinetically with the consideration that blood flow is responsible for distributing drug to various parts of the body. Uptake of the drug into organs is determined by the binding of drugs in the tissues. Unfortunately, this is a type of model that requires a very abundant amount of data, and it's usually reserved for research settings. Because of the amount of data and the complexity of this model to be used in practicality, these are not used in clinical situations. So this is how a physiologically based model is used. There is a blood flow, other parts of the body, and how blood flows from one part of the body to another. And that is it for our drug sampling and pharmacokinetic models. I hope you understood something. If there are any questions, kindly leave them down in the comments. You could read the book again, our Applied Biopharmaceutics and Pharmacokinetics by Shargel and Yu. This is a very great resource for understanding better the concept of drug concentrations and pharmacokinetic modeling. Okay, that's all for today. Thank you for listening and have a good day.